Let me know when it starts. Okay.
Yeah.
Okay, I think it's being recorded. Okay, so hello everyone. I am Sara, and together with Swagatama, we will moderate this EV characterization series talk. So today we have Ingrid Bloss, who will talk about innovations in EV characterization with nanoparticle tracking analysis. Ingrid Bloss studied microbiology and biochemistry at the Technical University of Darmstadt in Germany. For decades, she has been working as cell manager for lab machines and complex products in the medical industry. And since 2020, she holds the position of sales manager for Germany and parts of Europe at Particle Metrics. So today in her talk, she will first introduce the technology NTA, and she will present innovation and extended possibilities, particularly regarding the possibility of using fluorescent markers on the particles, on EVs, and colocalize them like on single particles. So I think that's all from me. And before I leave the floor to Ingrid, I would like to remind the audience that they can ask questions at any time in the chat. And we usually handle all of them at the end of the talk. So yeah, just feel free to ask questions and then we will read them at the end and we will start our discussion. So then Ingrid, the floor is yours. Thank you so much.
Thank you for the introduction, Sara. Let me see if I can share my screen here. Yes. I think you see the screen now?
Oh, yeah, okay. Yeah, we can.
Yeah, sorry. That's the end of the presentation. Okay, so hello everybody from my side. My name is Ingrid Bloss and I work with Particle Metrics in Germany. And today I want to talk about NTA technology. I have been informed that a lot of people are not familiar with the technology, so I also will talk a little bit about how do we do the measurements and what's the technology behind it. The title of my talk is it's more than just size and concentration, because if you look at the papers that are published now, which are using NTA machines, most have only used it to confirm size and concentration of the nanoparticles. But in the meantime, we can do so much more. And this will only come in the next years, I think, because there's always a time lapse between a new technology and when will the technology be published in the scientific papers. So what are the typical applications of the CW or the NTA machines? It's mostly bio-nanoparticles. That's the main application. So among them, extracellular vesicles or virus-like particles, viruses, liposomes, micelles, nanobubbles, but also inorganic stuff like nanometals or sorry, or polymers, emulsions. So you can use it for all kinds of technical applications as well. Basically, it works for all particles in the nanometer size range. Nanometer meaning from 1 to 1,000 nanometers, 1,000 nanometers, 1 micrometer. So how does it work, or how is it operated? It's very easy. You inject a sample in the machine, and then you immediately see the particles on the screen. So you see the individual particles. It's not only a curve or a value, so you can really see the particles, and then you can perform measurements. And the setup of the instrument is that you have laser illumination. And the laser is coming from the top at the right angles to the measuring cell. In the measuring cell, you have your nanoparticles dispersed in water, in buffer, or whatever medium, liquid medium you choose. And you can have 1 up to 4 illumination lasers. So you can have different excitation wavelengths of the lasers. And at right angles to the laser is a 10-fold objective, microscope objective, and a video camera behind it. And so the system is recording videos of the particles, and using these videos, to calculate certain parameters, which I will show you in a second. And also between the video camera and the sample, you can slide in a fluorescence filter, meaning you can also work with biomarkers to characterize your particles even in more detail. So the laser and the camera are focused, and then they are moving as a whole system together through the sample cell and taking readings at 11 positions within the measuring cell. And this means you get a rather large measuring volume, so you get good statistics. So you can you get a representative sample result, and you can analyze approximately 2,000 particles per minute. And you also have an included outlier control. So why do we do this 11 position measurement? Let me see if I can. Yeah, okay. Why do we do this 11 position measurement? This is because if you only check your particles at one position, you will maybe see only a very small amount of particles. Like in this example, you can see on the y-axis you have the absolute number of particles, which is here under 10 particles per peak. So this is not representative from your whole sample. And you cannot see, is this the real particle size? Is this the real size distribution, or is this just an artifact? But if I add more positions, I get a higher number of particles, of course, and I get a good size distribution curve. And I can also see that, for example, here in position 9, you see this big white blob, which is on the lower right side of position 9. This is an outlier. This is something else. It's either an aggregate of proteins or salts, or it's a dust particle or something like that. And the system will automatically recognize it and will filter out these particles, or will suggest to you to exclude them. In the end, the decision is up to you, because you are the scientists. You can decide how to process your data. But there are some algorithms which suggest you that this is probably not part of the sample you want to see, and it suggests that you exclude it. So what can I measure with NTA? We already talked about size and concentration. You can also, with our machine, you can also measure the zeta potential of a sample. You can detect subpopulations. In fluorescent mode, you can detect biomarkers. And you can also detect colocalized markers, which are on the same particle. And now I want to go into detail with all these individual points. So let's start with the zeta potential. This is the thing which is mostly for biologists not very well known, but zeta potential is the electrical potential at the slipping plane of a particle. The slipping plane is the plane where the charges go between mobile and attached to the particle. So so you can see here that there is a transition zone between the charged the fluid is directly attached to the particle, or it's moving around freely in the buffer or in the water. And the zeta potential gives an indication about the stability of a sample. So the higher the zeta potential is, the more stable your sample is, and the less prone to aggregation. So it's also, in the biological sense, zeta potential is also a valuable source of information which characterizes your samples. And here you can, for example, see if you have different zeta potentials, that it also influences the hydrodynamic diameter of your particles. So if the zeta potential is bigger here, meaning the minus 18 millivolt zeta potential will lead to a bigger particle. So this also has an influence on the size of your particles. So how do we measure the zeta potential? You have already heard that we are recording videos. And for zeta potential, we are putting an electrical field onto the sample. In this case, you see it's an alternating field, switching the polarity every few seconds. And this is recorded in the video. And the particle migration is captured for each single particle, meaning the system is tracing the particles over a series of images in the video. And then the electrophoretic mobility is calculated from the particle migration. And there are different approaches or different equations you can use to calculate zeta potential. Our system is using the Helmholtz-Smoluchowski approach, where the zeta potential is then calculated from the electrophoretic mobility. Concentration is measured or is determined by counting the particles, basically. So some of the video images from the video are taken and the particles are counted. And since we know the size of the field of vision and we know the focus depth of the laser camera system, we know exactly the measurement volume. And this is per position. It's about 3 nanoliters, meaning with the 11 positions, you have a total volume of 33 nanoliters, a total measuring volume. This sounds very small, but if you compare it to systems that work, for example, with a capillary flow, it's much more, because with the capillary flow, you would need a long time to get these 33 nanoliters. So the measurement time is much longer, and yeah, and there are several disadvantages in this, which I will also show you later. And we have found out that there is a linear relationship between the number of detected particles, which you see on the graph on the right-hand side, on the y-axis, versus the actual concentration of particles in milliliters. And this it's linear between 10 to the power of 5 up to 10 to the power of 9 particles per milliliter. So these are the this is the optimal concentration range to use for this NTA system. And this is not a very high concentration, which is fortunate, because if you are using bio nanoparticles, if you're using your real life samples, depending on the method how you harvest them, sometimes a high concentration can be a challenge. And if you have a high concentration, you can always dilute, but to concentrate your sample more is much more difficult, or sometimes even impossible. And the size measurement, it's all done on the same particles in the same cell, and you're also recording a video. And it's done by analyzing the Brownian motion of the particles. What you see here in this video, this is colloidal gold, 40 nanometers in size. And you see this, the smaller a particle is, the larger the Brownian motion, the quicker it moves. And you see this shivering of the particles, this is the Brownian motion. And since 40 nanometers is pretty small, they are moving pretty quickly. And the system is in a sequence of video images, the system is detecting and tracing the particles, meaning it's recording the movement in X and in Y direction. And from this, this is the so-called mean square displacement. And from this, and with the use of the Stokes-Einstein equation, you can calculate the radius of the particle, because in the end, the radius is the only unknown in this equation. You can analyze subpopulations if you have the right software for that. You can, for example, use a mix of different sizes. In this case, it's polystyrene beads, so plastic beads, basically, from size from 20 to 1000 nanometers. So this is a mix of 3 different sizes, and you get clear peaks of those. So you can analyze also different sizes of particles, and you can also analyze inhomogeneous samples. Here, for example, we have a mixture of plastic and silica beads, and the plastic is scattering the light a little bit more brightly than the silica, and therefore between these two, if you look at the graph, what you see here on the y-axis, it's the brightness of the particle, meaning the sorry. It's the power of the particles to reflect the laser light, or the amount of reflected laser light. And on the x-axis, you have the particle size. And they are both the same size. They are both 140 nanometers, but still, with our analyzing software, you can clearly identify 2 populations here. So even in scatter mode, you can already do some characterizing of your sample. But to go a step further, fluorescence would be is the key to characterizing your particles. So if you have your image like that, or if you look at your sample, the question you have to ask yourself is my particle really an EV or an exosome or a bioparticle? Is it really what I am looking for? Or is it maybe something else? Because all particles will reflect the light, the laser light, in scatter mode, and you will not be sure what kind of particles am I looking at here. And you will also not be sure if the size that you see is really the size of the particles you are looking for, because this is the total amount of particles. It could be anything. The solution to this is use fluorescent markers. So with fluorescence, you're sliding in a fluorescent filter in front of the camera, and then you will only see the particles which are equipped with these fluorescent markers. And you then can calculate the ratio between the total number of vesicles you see in scatter mode and the number of fluorescent particles you see when you're using the fluorescent mode. Meaning then you can also conclude about the quality of your sample. So here we have an example. This is a membrane staining where we used, I think it was cell mask green or cell mask deep red or something. So it was a membrane stain. So everything which has a membrane will be stained by this biomarker. And then we did a measurement in scatter mode, and we did a measurement in fluorescent mode. And you can see that we have approximately a ratio of particles which have a membrane of about 85%. So that means that 15% of the stuff you have in your sample has no membrane. So it's not a vesicle. It's something else. It's maybe some inorganic trash or whatever else is in your sample. So now in this step, you already see this is already the first information that you get. Okay, 15% of my particles have no membrane. So this is not what I'm looking for. I'm probably looking more at the other 85%. And if you want to go more specific, you can go into antibody staining. So in this next example, this is an example from practice we did a triple staining with typical exosome markers, CD9, CD63, and CD81. And they all were marked with Alexa Fluor 488. So they all have the same marker on them. So in this step, I only look at, do I have these exosome markers on my particles, but I do not look at the distribution or the percentage, how they are distributed. And I see here in the size distribution that there is a smaller amount of particles which are carrying these antibodies than the total number of particles again. So also you can see that the particles are smaller, the fluorescent particles, than the total of particles. And you can see that you have a lower concentration. So in this, so now you have done this triple staining, and now you see, okay, now I know even more about my particles. I know that I have only, I did not take the numbers here, but now I have only 6 times 10 to the power, no, it's 10 to the power of 6 particles versus 10 to the power of 9 particles in scatter mode. So I can see the percentage is smaller. And if you have more than one lasers in your instrument, like in this example, that's a twin system with 2 excitation lasers of 488 nanometers and 640 nanometers, then you can go even further and do a measurement all in one. You can do this membrane staining and the antibody staining in one go. So you can do this in the same sample. And you do not need to change anything on the system. You just leave your sample in, you press, it's one mouse click, and then you switch to the next laser. And you can do both measurements on the same sample in one go, and it only takes a few minutes. And so here you see that, as to be expected, there are fewer particles. When you look at the membrane staining, which is the red curve, there are fewer particles than in the scatter mode, and there are even fewer particles which have the antibodies. So it's about half of the amount of the total particles which actually carry these exosome markers. So only 50% of my particles are exosomes. And you can then proceed, if you have 4 lasers, you could then proceed to the next step and say, okay, I want to see how are these exosome markers distributed? How many CD9, how many CD41, CD63 do I have in my sample? And for this, you could actually label the antibodies with different fluorophores and measure them at different wavelengths. If you only have one laser, you can do all of this as well, but in more steps. You cannot do it in one measurement. You need to do 4 different stainings, and you need 4 measurement steps. But basically, it's the same thing, and you can do this if markers are available for the antibodies. Then it's also possible to do it with one laser. There are many available fluorophores. This is only this is not a complete list. This is the things we have already been working with and we have some experience with. But basically, especially for 488, which is a very typical wavelength also for flow cytometry, there are lots of markers available. So you can probably use a lot more than what we have on this list. It's just an example. So if you only have one laser and you still want to have a distinction between different markers, if you choose a combination of filters and the right markers, you can also do multifluorescence with only one laser. In this example, we did a staining with Alexa Fluor 488 and Percy-P. And both of these have excitation wavelengths, which is around 488. but Percy-P has a much larger Stokes shift than the Alexa Fluor 488, meaning that its emission maximum is much higher. With Alexa Fluor, the emission maximum is around 550, I think, or 520, and with Percy-P, it's I think it's 680 or something like that. So you see that this is emitting at a higher wavelength. If you only use one filter, a 500 nanometer long pass filter, meaning it will open up at 500 nanometers and let's pass all wavelengths which are larger than 500 nanometers, you will detect both. You cannot distinguish between both. So you can see Alexa Fluor and Percy-P. We get them both as positive. If you're using a multi-filter system with one excitation laser 488, and then using a bandpass filter, which opens around, or which goes around 550 nanometers, you will only detect the Alexa Fluor positives. And if you use another bandpass filter, which is around 660 nanometers, you will only see the Percy-P positives. So you can actually decide with only one laser. You can in one sample you could distinguish two different markers here. Of course, the range of available markers with different Stokes shifts is not as large as if you have more lasers. It's much easier to find markers, but there are quite some available with larger Stokes shifts, which you could also use with a one laser system. Yeah, one thing with fluorescence, maybe you have already worked with fluorescent dyes. One thing is, or one problem you might have is bleaching. There are some fluorophores which are very unstable, and some are medium to low stable, and then there are others with ideal photostability who will, if you look at the green curve here, they will not lose much information over the course of maybe 25 seconds. However, that is rarely the case. Most of them are medium or even low photostability. If you take FITC, for example, which is a very popular fluorophore, it loses half of its fluorescent power within half a second. So it's really not stable. So if you have a system which takes a long time to measure, like for example, all the systems which work in capillary flows, the fluorophores will just bleach before you have the possibility to detect them. So if you check at 0.5 seconds, which would be the bleaching rate of FITC, for example, you have with the medium to low photostability, you only use 93%. But if you go to 20 seconds, which is a standard measuring time of standard NTA systems, you only have 42% of the total yield of fluorescence. So you lose almost 60% of the information. And then the whole system is just not working, because you will never know the ratio between your scatter measurement and the fluorescence measurement, because this is so far away from the real value that you can never say. You don't know how quickly is it bleaching, and could I maybe calculate this, or it's impossible. So what you need is a really quick system. And what we did with the CW is we have the so-called anti-bleach or low bleach technology, where the total time of illumination with a laser is minimized as much as possible. So we have around half a second laser illumination of the sample if you're in fluorescent mode. And this means that you get the maximum possible amount of fluorescence signal. So in this comparison, you can see with the low bleach technology, the green curve, in 1 second, you do not lose very much information here. And with the standard NTA, you already lose a lot of information, and with the capillary flow NTAs, you lose almost everything. So it's basically, it's not possible to measure fluorescence and capillary flow. So then another part of my talk is also to show you what's the latest in NTA and what can you do with the latest models of these systems. You can do colocalization. This means I have nanoparticles which have 2 markers of which are excited by different wavelengths and which also emit at different wavelengths. And I want to see which markers are actually present on the same particles. And this can be done with the colocalization. Oops, sorry. So the colocalization will detect if the 2 markers are present on the same particle. So if you look at this little animated graph here, what the system is actually doing is it's illuminating the sample at exactly the same focus depth with both lasers. And it is switching between the 2 lasers and the 2 filters very quickly and taking a video. And then the software is checking if 2 signals of the 2 wavelengths are close enough together, then it will conclude that they are on the same particle. And so you can get a so-called colocalization ratio. And this could, for example, be done here. We have here 2 different membrane dyes on liposomes. Here you can do a colocalization, and for this is liposomes and membrane dyes. You get very good fluorescent results because the membrane is around the whole particle and it will include a lot of the dye. If you have antibody dyes, however, how many tetraspanins are present on your EV. It depends. The amount of fluorescent signal depends on the amount of antibodies which are even available. So in the first step, we started out with membrane dyes and with liposomes here. And here you can see that we achieved a colocalization ratio here of about 75%, which means 75% of these particles have the red and the green fluorophore on the same particle. But you can also go more specific. You can do, for example, a colleague of mine did RNA and membrane staining on phages. And he had very good results with the RNA staining with cyber gold. This is the dye. And Cellbrite 640 for the membrane dye. And here he actually achieved a colocalization ratio of about, I don't think that the 58 are correct. If I look at this picture here, I have around, it's around 40, 45. So this is, they are also always put in relation to each other. So if you have a higher concentration of the cyber gold and a lower concentration of Cellbrite, then you will have a lower, you will look at the 43. And if you take it the other way around, you set the Cellbrite as 100% and the 40 and the cyber gold as the relation, then your ratio will be a little bit smaller. So I think the 58 here is wrong, the number. Don't take me about that. Yes, so in a nutshell, I hope that I was able to give you a little bit of an overview how this technology is working. And to sum it up, you can measure a size range of around 20 to 1000 nanometers in fluorescence as well as in scatter mode. It depends a little bit on the light scattering properties of your particles. If you have something like gold, for example, which is reflecting a lot of light and it's scattering very brightly, then you can go down to 20 or even maybe even to 15 nanometers. For bio samples like EVs, for example, I would say it's realistic to say 40 nanometers is the smallest which we can get at the moment. And of course, also with fluorescence, it depends on what kind of marker are you using and what kind of and how much of this marker is present on, on the particles. You get a visual representation of the sample. You have seen the pictures of the particles in the system. So you really see the individual particles and the particles are not dried or immobilized, so they are just dispersed in their natural environment, in buffer or in water. So this will not falsify the results, and you will not get any artifacts due to the preparation of your sample. You can switch between lasers. So this means you have your sample in the cell and you can measure it with all available lasers. So you don't need to do any readjustment, any recalibration or anything like that. You can just click with a mouse and have measurements with all the laser wavelengths. The anti-bleach technology helps you to get as much fluorescent signal as possible. So it will give you a better yield of fluorescence, which is especially important if you're doing antibody stainings. If you are working with prefabricated plastic beads, which are already dyed with fluorophores, these will be very bright, and this will maybe also work without the low bleach technology. But if you're working with antibodies where you only have a very low signal because there are not so many tetraspanins on your EVs, then it's really, really important to use this anti-bleach technology to get back as much signal as possible. And another good thing is that that saves you money, is you don't need any expensive consumables. It's just, you just inject the sample with a syringe, and you need your tubes, and you need your pipette tips, and that's it. So you don't need any chips or anything which is expensive. And all CW systems are upgradable to 2 or 4 lasers, or also you can also upgrade to the colocalization feature if you already have a system. It would be possible to upgrade it. Yes, so that was that. Thank you very much for your attention. I want to point your attention to our cooperation partners who have helped us a lot and who are working with us, doing some application examples and doing a lot of research with our instrument. And so we are happy to have these people to cooperate with us for in order to also help all the other users to streamline the settings for the instrument and get valuable results which you can use in your own research. So if you have any questions, go ahead.
Okay, so we have a question here, and the question is that how many particles per frame should you try to visualize, and what's the colocalization analysis, and like at what point do we do that?
Yeah, particles per frame is, yeah, you have, if you are familiar with the software, you will see there is an indication of the number of particles that are visible, and you should go between, I would say, 150 to 200. It's the same as with scatter measurement, and well, with fluorescence measurement, it's probably sometimes a little bit lower, but it's possible to also do with a lower amount of particles.
Okay,
Thank
so
you,
we do
Jenna.
have a lot of we have a lot of positive feedback from this session, because I mean, well, NTA, especially particle metrics, has done a great job in this field, and I have used the particular equipment last year. So we had a session here in a university, and I actually had the pleasure of listening to Ingrid twice. So you were the one who actually helped us do this session.
Oh yeah,
in a
I
virtual way.
recall. Yes.
Yeah, so that was very, very amazing. And one feature I'd like to really point out for newbies like us who are starting off in this field is that when the analysis report comes in the zeta view, there's this one measurement in which they show the different diameters of all particle sizes. So it's not like they're gonna group up particles in specific ranges. They'll actually give you a detailed interpretation of what all particle sizes were recorded and what's the percentage concentration of it. So it gives a very clear view of what kind of let's say clumps or what sort of EVs you're looking at. So that was very helpful for me. And yeah, so we have a question with saying that, do you think it's necessary to use Q dots to analyze fluorescence, or is it the antibody alone sufficient?
Well, it's hard to say, Sergio, because it really depends on if you have enough antibodies, if you have enough tetraspanins on the EVs. It's sometimes the distribution is different. If you're using the typical markers like CD9, CD63, and so on, the distribution depending on the origin of your particles is different. Sometimes you have more CD9, sometimes you have more CD81. So if you just want to make sure that you have EVs, then I recommend this triple staining I showed you, that you have all three markers with the same fluorophore. But if you want to distinguish between the individual markers, it may be that if one of them is not very present on your sample, it may be that the signal is too low in this case. Okay, there's another one about fluorescence antibodies. Oops. Okay, let me see if I can find this slide again. I'm just going to show you the screen.
Also, yeah, we will be recording the session. It'll be available on SNEV's website. And apart from that, we also, you'll be able to have access to this entire session over there. And I'm not sure if we can have the detailed explanation right now, but I'm sure we can expect future sessions to.
Yeah, I'm just not sure which of which we are talking about which video. So that was a little bit.
I think the copy of this session, I mean, there's this entire meeting. I think that is the one Jana is asking, if I'm not wrong. And yeah, then we have another question. How much volume do you need to run one measurement? And for EVs, can you measure accurately the larger sizes, like 500 to 800 nanometers? Because with nanosite and other NTA systems, these sizes are not entirely accurately detected, even if these sizes are in the detection range.
Yeah. So first part of the question, you need 0.5 milliliter, 500 microliters. That's the volume of the cell. However, we recommend to aim for 1 milliliter because to make sure that the whole cell is completely filled with sample, it's better to inject 1 milliliter so that you're sure that also in the outermost positions of the cell, there are enough particles that you get good statistics. And the other one is, yes, you can measure larger sizes like 500 to 800 nanometers. However, you have to adjust the settings. If you're expecting something like that, the larger sizes, you have to adjust the settings, the SOPs of the system. But we give you a number of sample SOPs, which you can take as a starting point for your own particles.
Thank you. I think, Maimona, you said like you don't hear Swagatama well. So maybe I will continue reading the questions, right?
Mm-hmm.
Yeah, so actually, Ingrid, yeah, thank you so much for the talk and for all this information. And so because I asked the question about the size, and I wanted to ask, like, so for example, if you have a sample that is heterogeneous, in particular like EVs that go from 100 nanometer to 800 nanometer, can you measure those at the same time? Like, can you see the size distribution? For example, if there is a peak at 200 and a peak at 700, can you see them in the same measurement, or would you need to run two separate, two measurements with the sample and adjust the parameters for each part?
If it's a really widespread, like you go from 50 to 800 nanometers, I would recommend to maybe two runs with different settings to be sure you catch them all. Because you just need for smaller particles, you need, for example, a higher video resolution, because a lot changes between the individual images if the particles are moving quickly. But if you're having larger particles, this is counterproductive, because if nothing changes between 2 or 3 images, the system can no longer calculate the size. So you need a lower resolution for the bigger particles. So if you run maybe with 2 settings in the same sample, then you get a better representation.
Okay, great, thank you. Now I will continue with some other questions that we have in the chat. So one person is asking, so as to get an accurate measurement of size distribution and concentration, is it recommended to measure different dilutions of the same sample, and in which buffer should we do the dilutions?
Okay, you don't need different dilutions to get an accurate measurement. The first thing you would do if you have an unknown sample and you have no idea about your concentration, I recommend you do a series of dilutions before you do the first injection, and then you start with the lowest concentration and work your way up until you have reached approximately 150 to 200 particles on the screen, and that's the optimal to measure the concentration. You can also enter in the system, you can enter the dilution factor. For example, if you did a dilution 1 to 100,000, you can enter this, and the system will also calculate you the original dilution, the dilution of your original sample. And you can do it in whatever buffer you want to use. The system has 2 liquid systems. One is water, and the other is your buffer of preference. If you want to switch between buffers, between measurements, the only thing is you need to rinse the system for a few seconds to equilibrate it, so you don't have any turbulence due to different charges on the buffers.
Okay, great, thank you. Another person is asking, first he says, thank you for the great presentation. May I know what are the parameters do we need to report when we use the scatter setting? Another question, do we need to calibrate the NTA machine every time before we use it?
Okay, second question, you don't need to calibrate. What you are doing is when you are setting up the instrument, when you're starting it up, when you want to use it, the system recommends a focusing procedure, meaning you inject some sample beads on 100 nanometer polystyrol beads, and the system uses these to get a sharp image. So it's moving the laser and the camera towards each other until the image is sharp. It's not a calibration per se, but it's a focusing process. And I am not sure about the parameters. If you are referring to sensitivity and shutter, that depends on the sample, but I assume that you are using EVs. And for this, we have some sample SOPs. We have some SOPs we give you. I cannot tell you the values now because it depends a little bit on your model. It may be different for different, but the sensitivity and shutter settings are in the SOPs, which you can use as a starting point.
Okay, great, thanks. And I think the last question is actually similar because it's again about the calibration, but the person is also asking if, okay, now there is another one. So if also when you change between materials that have like very different intensities, if you need to do any calibration or not.
Okay, the calibration particles are polystyrene, what we are using. However, it's true, EVs have a different scattering intensity. They scatter light a little bit lower than the polystyrene particles. If you wanted to calibrate or to focus on particles with the same scattering properties, you would have to use silica beads. However, these are much more expensive and much more unstable. So this means it would raise the working costs of your instrument. And we have worked out the SOPs in a way that you do not need to worry about this discrepancy. So if you do the focusing with the polystyrene beads and you're using the SOP for EVs that we give you, you can be sure that it's the correct concentration. And okay, so you're working with Q dots, I see. To tell you the truth, I have never worked with Q dots, so I do not have any experience with that. I would, however, offer to ask my colleagues and then maybe to write to my Mona, and she could maybe distribute this information to you, because I know that one of my colleagues has already worked with Q dots, so I can maybe give you some hints after this presentation.
Yes.
And yes, the last question
for publication, I see. Yeah. Okay. Yeah. Well, you can use all information you get from the system. You can have the size and the concentration. You get these curves that you have seen in the presentation, something like this, which you can also use for your papers. And you have to indicate, yeah, you can. I think you can look at our website. We have a link. We have links to many publications which have worked with our system. If you want to know how to cite it in the material methods part, you can check there what people are doing. But most are only writing NGA system by particle metrics. They are not even talking about the model or something like that.
Okay.
Yeah, I might have one question. Yeah. Yeah, thank you so much for the interesting talk and yeah, for the answering our questions. So yeah, I'm asking a question about the beads. For you said it's for focusing, it's not for it's like calibration, right? Yeah. So if we are using smaller beads like 20 nanometer or something, will it affect the reading? Because like in my case, we're getting the reading for our EVs is very small, and I discussed with the professor, and he suggested maybe they're using a very small beads to adjust the focusing. Yeah. So
no, it will not. It will not make a difference. You can, if you want to be absolutely sure, you could use 20 nanometer gold particles to do the focusing, but the SOP and the autofocusing process is based on these 100 nanometer polystyrene beads, and it's only to make sure that you have a sharp image, and this is also working with the polystyrene beads. So you don't need to get anything more expensive and more complicated. It should be okay. It's maybe more the settings if you really want to see 20 nanometer. Yeah, it's. Do you think that you have 20 nanometers in
No, we're getting
EVs?
them at 60 nanometer. Our EV size is 60 nanometer. So he suggested that this is too small for EV and for our MSc EVs. And then he suggested maybe this is because they're using beads that are of small size of 20 nanometer. That's why we're getting a very small. So
I see.
It's not accurate.
No.
Yeah.
No, I don't think it affects the size measurement at all. And if you want to be sure, try some antibody staining and then you will.
Scattering, we don't have actually a fluorescent one. Yeah, I would love to work fluorescent, but this is for scattering, so we can only do size. Yeah. Okay. So thanks for answering my question. Yeah.
I have one question. Maybe if we have like 4 more minutes left. That is about the fluorescence mode. So if you have a system with like 4 lasers that can measure 4 channels, do you have to pay attention on the fluorophores you choose, or like, because could you have crosstalks between the different channels, or you are free to choose whatever fluorophores are available and like the system will not mix this channel?
Well, it will. You are worrying about spillover between between the wavelengths. So they are actually pretty far apart. It may be between 488 and 520 are the ones which are closest together. There it depends a little bit on what kind of, if you have a really bright fluorophorescent, really a lot of it, it could maybe reach out into the 520 channel as well, but we rarely see this because mostly the signal is not so bright and not so big that you get a problem with spillover. And for the colocalization, we are using only the combination for 88 and 640 at the moment to avoid this spillover effects completely.
Okay, nice. Okay, thank you. Well, I think that, yeah, we read all the questions in the chat, and yeah, we're almost at time. So I think we can thanks again, Ingrid, a lot. This was very interesting. We had a lot of, yeah, we had a lot of participation and a lot of questions. So that was great. Yeah, so I think we can stop recording the talk, and as Swagatama said, this will be this will be posted in the YouTube channel of SNEV, so you can rewatch it there. Thank you also, Ingrid, for allowing us to record, and yeah, for the very interesting talk. So yeah,
okay.
I think we can.
Thank you very much.
Here.
Thank you.
Thank you also in the chat. Thank you.
Thank
Have
you.
a nice day. Bye.
Bye. Thanks for having me.